Good evening, everybody. Welcome to, I guess this is our fourth One Health seminar of the semester. For those who have maybe not joined us in the past, let me tell you a little bit about One Health. So One Health is a global concept. It kind of gets approached in different ways, but here at DelVal, we are approaching One Health as being a systems approach and recognizing that people, plants, environment, and animals all share this planet together. And frankly, I don't care what major our students might be in or what kind of field you work in. You probably have already come to the awareness that you have to work across disciplines, and that just because what you learned in school may be helpful to you, you always benefit from listening to other perspectives. So the One Health is really this transdisciplinary systems approach, looking at problems from a the need for transdisciplinary kind of interactions at the local level, the national level, internationally as well. And this works for a wide variety of issues. And so at DelVal, instead of looking at purely from a health perspective, certainly we recognize that COVID-19 is where there's a nexus between disease, animals, and people, and our societies and how we deal with wildlife and other animals. Uh, we believe that this really fits for all majors. And so we kind of approach this from three different perspectives. One is education. And so with education, we're really looking at it within individual coursework. We actually do have a One Health transdisciplinary degree that's coming along that will be offered. But really, I think it's pretty clear that no matter what your major is, there are interactions with environmental and animal issues. And so why don't we recognize these as really being a One Health kind of approach? We also are fostering research. We would like to see research across disciplines to deal with some of the problems that exist not only locally, but nationally and internationally. And then tonight is all about outreach. And so we offer these 6 seminars each semester, twice a year. Uh, and we also are welcoming to having anyone who's interested in this concept to kind of join us. And that is where our co-sponsorships come in. And we frequently will have co-sponsors for our One Health seminars. And somebody who won't be much of a stranger to most of you, if you've been listening to any of the One Health seminars, we have Shannon with us this evening from the Heritage Conservancy. This seminar tonight, as it is with most of our seminars, they are recorded. And so if you're interested in seeing past seminars or seeing this one later on, sharing it with a friend, uh, don't hesitate to go to www.delval.edu/One Health, one word. And now I'd like to introduce Shannon from Heritage Conservancy.
Thank you so much, Reg. I'm glad that we could be here as co-sponsors again tonight. I'm Shannon Friedeboss Stiller with Heritage Conservancy, and some of you've heard probably some of our spiel, but I'll add some new highlights as I try to each time we co-sponsor. So Heritage Conservancy is a local nonprofit, and we serve Bucks and surrounding counties here in southeastern Pennsylvania. Our mission is to preserve and protect our natural and historic heritage. Since 1958, we've assisted in the preservation of over 15,500 acres of open space, farmland, wildlife habitat, and important watershed areas throughout the region. So we're excited to co-sponsor tonight's presentation, not only because of the importance of the topic about this prominent native tree, the American chestnut, but also because there is a research orchard on our Fuller Purcell Nature Preserve. This preserve is over 100 acres of contiguous forest and is located in the northern portion of Bucks County. The picture here that is on the screen shows the installation of the American Chestnut Orchard, which took place in 2002. Heritage Conservancy, the American Chestnut Foundation, the Cooks Creek Watershed Association, and Bucks County Boy Scout Troop Number 52 planted 50 American chestnut seeds, including pure chestnut seeds and hybrids that are partially blight resistant. So I'm not going to go into that any further because I know that's what Mary is going to be talking about tonight. And I'm sure she's far more knowledgeable on the chestnuts and the work to try and combat the blight impacts. Next slide, please, Reg. And on this next slide, so this is actually a photo of the orchard right now. And this is what a volunteer was able to take a picture of last year while he was at the Fuller Personal Nature Preserve. You can see the protective deer fence is still surrounding it, and a variety of the chestnut trees are doing very well up there. So feel free to visit our website, heritageconservancy.org, if you would like to learn more about the Fuller Personal Nature Preserve, or if you'd like to visit the hiking trail there. Our website is also a great place to learn more about how you can join in our mission through events, membership, and volunteerism. Plus, we have a few job openings, especially summer internships available right now. Heritage Conservancy is delighted to co-sponsor this evening's presentation and welcome Mary Ayers with the American Chestnut Foundation.
Hello, everyone. My name is Mary Ayers, and I just need to get my screen sharing. There we go. I'm very pleased to be here tonight to tell you about the American chestnut and why we feel it is so important to restore this tree to our eastern forests. Now, I just want to have a very brief disclaimer up front that I am not a scientist, I'm not a forester, I'm not trained in environmental sciences. I'm just someone who likes trees and forests, and began to join the American Chestnut Foundation 11 years ago, and have been involved as a volunteer for the organization since then. Now, what I'm going to tell you about is basically the life, the death, and the rebirth, we hope, of the American chestnut. It's important to understand that this tree was an amazing tree. It was the king of the eastern forests. It was a keystone species in the eastern forests of the United States. It grew 100 or more feet tall, 15 or more feet in diameter at maturity, and it could live for 500 years. Now, keep in mind that a tree that's 100 feet tall is the height of a 10-story building. And fun fact, as an aside, in the state of Pennsylvania, the champion tree is a white pine up in Cook State Forest, and it's around about 180 some feet tall, which puts it at about the height of an 18-story building. These are the kinds of forests that we really don't see anymore, because most of the forests in the eastern seaboard have been harvested, some of them multiple times. So in the state of Pennsylvania 100 years ago, 95% of the tree was the state was deforested. So many of what the forests you see out there as you drive through the state are second growth forests. They're equal age forests, and they're not that old. I mean, mature forests will have, it was common for oaks to live 200 or 300 years, and as we said, chestnuts could live 500 years. So it was a very important tree, and it was in the range. You can see that highlighted here in the dark green. There were areas where 1 in 4 trees in the forest was an American chestnut, which is pretty amazing in terms of its how prevalent it was. Now, the tree was really, really important to the settlers and the population of the United States until it was killed off by blight. It was an important timber tree, a very important timber tree. It was primarily, it's a highly rot resistant wood. So it was primarily used for things like barns, for fencing, for poles, and think of the expansion that took place in the 19th century with the railroads, the telegraph, and later telephone lines. It was a very strong tree and very rot resistant. In fact, it's just slightly less strong than oak, but it weighs less than oak. And if any of you ever lifted oak, you know that if you're going to be building something, you prefer to not necessarily be building with something quite that heavy. So it was a very important source of tannin, which was used in the leather industry. Remember, back then, everything was horses, and there were bridles and saddles and all that stuff. Huge demand for leather. It was a really, really important food source, both for wildlife and for humans. The chestnut tree produces nuts every year. It was a reliable producer of nuts, and the nuts were very sweet, made a lovely flour, source of food for wildlife, for birds. And in Appalachia in particular, I mean, people ate chestnuts. It carried them through the winter in a lot of cases, but it was also a cash crop. And there were every fall, there were railroad cars full of chestnuts being sent to the major cities on the eastern seaboard for roasting over the open fire. So it was a really important tree and a very iconic tree, and has a lot of history in our culture. Now, so what happened? A chestnut blight came in, and they believed that it was imported on either Chinese or Japanese chestnuts. If you know anything about the 19th century, it was a time of tremendous exchange of plant material. There was a lot of plant expeditions taking place throughout Asia and Africa and South America. And it was all the rage to have exotic species and new species brought in and grown here in the United States. So obviously, people imported Japanese and Chinese chestnut. And in 1904, tankers were discovered at the Bronx Zoological Gardens in New York that were killing the trees. And gradually the blight spread. It took off. Back then, there were times in the teens and the 20s, Pennsylvania actually had programs to clear cut areas to try to prevent the spread of the blight, but there was no preventing the spread of this blight. And you can see from the map that by the in 1950, it had really reached the end points of the range down in Alabama and Mississippi. And it was pretty catastrophic. Now, since then, as you all know, we've had Dutch elm disease. We have emerald ash borer now, which is basically killing all the ash trees, and they're killed dead by the ash borer. And in Pennsylvania especially, which is a central part of the hemlock tree, the hemlock woolly adelgid is also hitting those trees. So our forests are under a lot of stress. But the chestnut blight was one of the biggest devastations that we've experienced in our forests. Now, this is a picture of the blight on a tree. It's called, the technical name is Cryphonectria parasitica. And what it does is attacks the tree through cracks in the bark or through wounds. It'll typically, a lot of times you'll see it in the joint where a branch comes off the stem of a tree. And it's these big orange cankers basically kill the cambium, which prevents the tree from getting both water and nutrients, and the tree dies as a result. Here's another picture of a chestnut tree. You can see that there's blight there on the main stem and also on these peripheral stems as well. Now, one of the interesting things about the blight, and this is important for the story of the restoration of the American chestnut, is that it doesn't actually kill the roots. It kills what's growing above the ground. So the roots continue to send out shoots, but whoops, why am I not moving forward? There we go. So as the blight spread, as a result, about an estimated 4 billion trees were killed by the blight. And it's very rare to see any sort of major size American chestnuts surviving in the forest. And those that you do occasionally find tend to be not to flower or fruit, because chestnut trees need a male and a female from a different tree in order to produce nuts. So it's pretty bad. Now we call this, so the tree is not technically extinct, but it's what we call functionally extinct. Because the roots keep pushing up shoots, you will find thousands. They estimate, well, according to the slide, Dalgish, whoever he is, estimated 430 million stems in the forest. If you hike along the Appalachian Trail, it's common to see chestnut stems. The problem is they don't grow to any size. 84% of them are 1 inch at diameter at breast height. It's not very big. The blight attacks the sprouts and they die back, and the roots, which got some energy pumped into them from the leaves of those sprouts, will send up more sprouts. At one time, Sarah Fitzsimmons, who's our director of restoration, said that although it's functionally extinct right now, and there's a lot of germplasm out there in the form of chestnut sprouts, in about 1,000 years, those they believe that the stumps that are out there from the original chestnuts that are still putting out sprouts now will no longer be doing so. So now you can find chestnuts primarily in late June and early July when they come into bloom. That's one of the best times to spot them, because that you will see the male flower, which looks like a pipe cleaner. And it's the female flower that turns into the burr that ripens, and inside each burr are typically, in the American species, 3 nuts. It varies. I think the Chinese has 4. I'm sorry, I'm not technically versed in Chinese chestnut. And the burrs are large, and they're very, very prickly and spiny, and you better have gloves on if you're going to pick one up. Now, there are major differences between chestnut species. The Chinese chestnut and the American chestnut actually split about 30 million years ago genetically. There are commonalities, of course, but there are differences in the shapes of the leaves, twigs, the shape of the mature tree, the nuts, etc. American chestnut has the smallest nut of all these species, and the leaf is canoe shaped. It's a distinctive shape of the leaf. The leaf is, I did a bit of training on identification of American chestnut versus Chinese chestnut. And let me tell you, I can't even describe to you how hard it was to see these things under a microscope or even an eye loop. The American chestnut leaf is hairless. That was the easiest identifying characteristic of the tree. The leaf is very thin and canoe shaped, and the very tips of the tree, of the leaf, are what they call, have a breaking wave formation, like an ocean wave that's breaking. And the twigs are different. They're hairless and red to chestnut brown in coloration, and the bud is very smooth, brown, and pointed. In contrast, the Chinese chestnut has very shiny leaves, and they're usually hairy on the underside. That was the easiest thing to tell when we did the leaf identification. The leaf is thick and leathery, it's not delicate, and it's more oval shaped with wedge shaped tooth margins. And the twig is more green. You can see here how green it is, and it's hairy, and the bud is round rather than pointy, and also green rather than red. So there are differences, and you might say, well, but what does it matter? I mean, if you know these things, that they're different in habitat and the environment and how they perform, why not just plant Chinese and Japanese chestnuts? Why not put them into the forest? Well, guess what? They're not locally adapted. The Chinese and Japanese chestnut trees are not forest trees. They're not big, single stem, tall, light-seeking trees that will thrive in the forest. Every year, our chapter of the American Chestnut Foundation gets about several hundred samples, leaf samples sent in because people want to know if they have a wild type American chestnut, and the vast majority of the samples that we get are Chinese chestnuts. They were distributed widely in the 50s and 60s by Penn nurseries, so you find them around the state of Pennsylvania. They're often, I volunteer at the Pennsylvania Farm Show every January, and I can't tell you the number of farmers who come up and say, well, we got a chestnut tree growing in the front yard, and it's almost certainly a Chinese chestnut that they have growing in their front yard. The Chinese and Japanese chestnuts just can't compete in the woodland. They don't have that straight, tall trunk that will seek the light when you get a forest opening, which is when trees really shoot up in a forest setting. And and mostly the Chinese chestnuts that you see out there have been bred for orchard production or as ornamental trees. So our goal is to return the chestnut tree to the east as a forest tree to the eastern forests of the United States, its native range. Now, the American Chestnut Foundation is not the first organization, and there's many people working on this problem. Back in the 1930s, the USDA and the Forest Service were all trying to breed blight-resistant chestnuts, and everybody is always hoping that they're going to find trees out there in the forest who are surviving without blight, and it's in fact pretty lethal. And yeah, there's occasional survivors that you can find, but more often than not, they get blighted and die, even if they did live for 20 or 30 or 40 years. Now, the organization we rely on a lot of partners, a lot of different universities. Our chapter is based at Penn State. The headquarters of the American Chestnut Foundation is down in, well, the offices are in Asheville, and the main research orchard is in Virginia. But we partner with the U.S. Forest Service, the Bureau of Mines, because chestnuts have been planted on mine reclamation sites. This is a sustainable forestry initiative, and ESF, I want to point out, because we'll be talking more about it in a little bit, is the State University of New York College of Environmental Science and Forestry. And that's where the work on the transgenic American chestnut's been done. So the organization began in 1983. We now have 5,000 plus members in 16 chapters across Eastern United States. And what the organization is doing is basically using everything in the toolbox that we can to come up with a blight-resistant American chestnut. Historically, the organization began with back cross breeding program. Think Mendel and the peas. Basically, you take a Chinese tree, take an American tree, and you cross them. You plant the seeds that come from those trees, and then you cross those with another American, and then you plant those seeds. And each generation took 5-6 years before the tree would grow up. It would produce nuts and so forth. And what they would do is inoculate the trees with the blight. And if the blight killed the tree flat out, well, then you wouldn't collect the seeds from that tree. And it's what they call selection. And but anything that showed some resistance would be carried forward to the next generation. So this is what the Chestnut organization did for years. And this is a picture of one of our orchards at Penn State. And the trees on the left are unselected, and the trees on the right were selected for blight resistance. And now, luckily, we now have, we're applying genetics to the selection, and we have things like small stem assays. So you can actually test a tree now during its first year to determine its degree of resistance. So we don't have to wait for the tree to grow for 4 or 5 years before you determine whether it's showing any resistance. The picture is Sarah Fitzsimmons. She's, again, the Director of Restoration for the American Chestnut Foundation, and she's headquartered at Penn State. But one of the great things to come out of the breeding program is that genetic analysis of the backcross trees, which have some Chinese genes in them because they're bred for resistance, is that we can begin to compare the genomes of the backcross trees with that of wild type Americans. And by looking at the differences, you can begin to locate where the resistance genes or alleles or whatever they are located. This is where I break down. I'm not a scientist, and I'm really pretty bad at talking about the genetics, the deep that's involved. So one of the things we've determined in the course of doing genetic analysis is that the backcross breeding program in and of itself was not going to give us a blight resistant tree that was 95% American genes with only 5% Chinese genes, because rather than there being 2 or 3 genes that confer resistance, which is what everybody thought back in 1983 when they started the program, there's at least 9 genes involved. And if you know anything about math, it's really, really hard to breed trees that would carry all of those 9 genes. And the other difficulty is that every one of these trees has to be bred with locally grown American trees, wild type Americans. Genetically, you need that diversity across the range. A chestnut from Maine is not necessarily going to grow well in Alabama or Georgia. So when we did the backcross breeding program, we didn't just have one orchard where we're doing this breeding. We had orchards all throughout the range of the American chestnut. At Penn State, It sounds like you've got the Heritage Conservancy has one of the backcross orchards, because you needed to have that local adaptation represented in the breeding stock. So the other another prong of the American Chestnut Foundation's 3-burr effort is biocontrol. This is my only slide on this. I'll try to make it brief, but basically, the idea is that if you can make the blight hypovirulent, meaning that it is less virulent, meaning that it would not kill the tree, you could have the blight, the tree could get infected, but it wouldn't die. So they've actually have worked on hypervirulence that's used somewhat in chestnut orchards in Europe, because Europe has the same problem with their chestnuts and having blight. They're susceptible to blight. So, but one of the difficulties right now is you can take this hypervirulent virus, the virus that carries the hypovirulence or makes the fungus less virulent. See, this is where I'm breaking down in terms of the science. You can do that, but you have to basically paint the cankers with it. So the issue is, and there's a lot of study being done on this, is how do you make it so that that would sort of spread out into the environment to basically transform the blight that's out there to be less lethal, and that they don't know how to do yet. Because think about it, you've got a tree that's 25 feet high and it's got blight up toward the top. Who's going to climb the ladder and paint that? You can't, that's not going to work in a forest setting. So again, an area of research, it's an important area to look at to understand how the blight works and this method of control. But right now, that's one prong of the research, but that's all I'll say about that. Now, biotechnology, you heard me talk about the transgenic tree. This is the third main leg of our approach to coming up with a blight-resistant American chestnut. Dr. William Powell, who's at SUNY ESF, in the early 1990s, was interested in this problem of the American chestnut, and he developed a lot of techniques for inserting the gene of from the wheat plant. It's called the oxo gene, that neutralizes oxalic acid that is produced by the chestnut blight fungus. So fungus infects the tree, it secretes the oxalic acid kills the cambium. So what he did was, this particular gene neutralizes it. It's found widely in nature. It's found in strawberries, it's found in wheat, it's found in a lot of plants in our world. We eat plants that have the oxo gene in it all the time. Anyway, he inserted this gene into an American chestnut, developed some, they're obviously clones, a transgenic chestnut. It's called the Darling 58. And it is currently under regulatory review by the USDA. Once it passes that, it has to be reviewed by the EPA and then the FDA. But we're hopeful that in a few years, it might pass all those hurdles and can be we can begin the outbreeding and introduction of the tree in the forest setting. Now, these trees are growing out there in the world, but they're not allowed to just have their pollen go free. So anytime the tree flowers, all of the flowers have to be bagged so the pollen doesn't get loose. or I think they cut them off, or they do whatever they do to inhibit that. Now, the beauty of this transgenic tree is that the OXO gene is a dominant gene, meaning that 50% of the offspring inherit it. So if you cross one of the transgenic trees with you outcross it to a wild type, locally adapted American tree, then you get nuts, and those nuts and you grow trees from those nuts. About half of those trees will carry the gene that prevents the blight from killing the tree. The other really good thing about this is that it doesn't actually kill the blight. As you know, with anything in genetic modification, I know a lot of people will think of things like glyphosate, where you now have glyphosate resistant weeds coming along. There's a lot of research been done, but it doesn't put as much pressure on the blight to evolve if the blight itself is not affected. So it just neutralizes the oxalic acid produced by the blight. So it doesn't actually kill the blight. So you get minimal amounts of pressure in that regard. So in the future, what we plan to do is outcross this tree with wild type, locally adapted American chestnuts, and outcross it with the backcross hybrids that we have in orchards around around the country. The importance of that is that you can get what they call stacked resistance. So in addition to having the OXO gene, some of the trees will pick up some of the Chinese genes that confer some resistance. They won't be, quote, pure American chestnuts, but they'll be very close to that. Now, as part of the regulatory review of the transgenic tree, they had to do a lot of studies, and this is an area that any of you in biology, conservation biology, ecosystems analysis, whatever, they really need people to do more research on, especially insects and their relationship with American chestnuts. But these are some of the studies they did. They did a study on leaf litter decomposition. I think they looked at whether worms decompose it at the same rate as they decompose Chinese chestnut leaves, germination of the seedlings from chestnut leaf litter, herbivory on leaves, colonization of mycorrhizal fungi on roots, which you know is super important in forests to make sure that you're not messing up that underground fungal system that trees depend on, bee feeding on the pollen, tadpole development after feeding on the leaves. These were just some of the things that they looked at. So how can you help, or how can anyone help? Obviously, our organization's always looking for members if you're interested. We're always looking for volunteers to do outreach. But the two key things here are highlighted in blue. We really, really want people to plant American chestnuts. And one of the reasons is that we need to have the genetic diversity. We want to have at least 3,000 unique sources of American chestnuts from across the range. And we're always interested in locating new specimens of American chestnut. There's a certain priority areas that are poorly represented. There's some areas like in central Pennsylvania that are well represented in terms of pure Americans. But this is what we call our germplasm conservation effort, because once we get an a transgenic tree that is blight resistant, it has to be bred with locally adapted trees. It needs to be outcrossed with them. There's a particular app that's available for iPhones and Android that it's called Tree Snap. And it actually covers several species that are threatened. and the data is funneled to academic institutions. And so if you're ever out in the forest and you find what you think is an American chestnut tree, you can geolocate it. It asks you all sorts of information. You don't even have to be in Wi-Fi. It'll gather all the information, and the next time you're in Wi-Fi, or if you're on a cell plan, it can upload the data that you collect. The key thing with American chestnuts, we're always interested in finding new sources, but they we really, really, really like to get a leaf sample, a leaf and twig sample, because that's the only way we can really tell if it's an American chestnut and not a Chinese chestnut, which, again, as I've told you, most people send in Chinese chestnut samples. Now, one of the problems and one of the important reasons for conserving the germplasm of American chestnut trees from across the range is that in their wild state, they're mostly not flowering or fruiting. It's very rare to find that. So we've got a big initiative to graft wild trees onto rootstock in order to gather the germplasm. And it's very interesting. What they do is they graft the trees. There's a problem with grafted chestnut trees. They tend to kind of drop dead after like 7 or 10 years. So what they do is they'll graft the tree, and they can force pollination in I think it's the second year of growth, flowering, and then they can collect the pollen. So then it kind of doesn't matter if the tree dies, they've got the pollen, and then the grafted tree, and then they can cross anyway, they can use the pollen off of the tree. So it's very complicated, but it works in order to conserve the germplasm. And then, of course, we love to collect seed, actual chestnuts from rare flowering trees, but it's rare to find flowering trees out there in the environment. So that basically brings me to the end. I wonder if you have any questions. I'll make my best effort to answer them. Again, I'm pretty weak on the deep science of it all, the genetics of the back crossbreeding. But before we go on, I did want to just mention one thing. I don't have any slides on this, but one of the challenges we face moving forward once you have a transgenic tree is, how do you actually reestablish it in the forest? And they're doing a lot of work on that, and a lot of research on the best methodology, but it could take, centuries for a few planted trees to, the flower and fruit and spread and so forth. Chestnuts typically need a lot of sunlight to grow to enough height to compete in the forest and begin to flower and fruit. So that means you have to have a clearing. There's a lot of acres out there. You would need a lot of seedlings. It could take thousands of years based on current seedling production to get the tree restored in the eastern forest. So there's a lot of challenges to actually taking it to the next step and ensuring that it's established in the forest and not just an ornamental landscape tree. So there's some of the issues we face, and it'll probably take 500 to 1,000 years before you start walking through the forest and seeing mature American chestnuts in any great number. And again, if you have any questions, and for more information, go to patacf.org or ACF or tacf.org. My bad. Reg?
Yes, so I am here. Thank you so much for this. And I really want to thank the American Chestnut Foundation for all that it's done to try to begin this process that, as you said, will take a long time for us to potentially have back a functional forest that includes American chestnuts. But I, for one, and I'm sure there's a lot of people on here tonight who would agree that it's well worth the effort to do that. Uh,
Yeah.
we have some chat and we have some Q&A. Let me read the chats here first, and then we'll get over to the Q&A. So within, actually, got quite a few here in the chat. So from Emil, I have several American chestnut trees, 2 old and 5 younger trees. All are nut producing. I am located in Buckingham Township. If you would like seeds, please contact me. And he gives his email address. And he continued that, giving his email address and mailing address. I guess you might want to ask him if these have been evaluated for actually being American chestnut trees.
Yeah, that's my question. If they have great size, they're flowering and fruiting, they're probably not American chestnut, and they're not blighted. They're probably not American chestnuts. But I can't say for sure, and that's where we would love a leaf sample. A leaf and twig. Don't send one leaf. You send like a branch with the leaves. And anyway, there's a tree identification form available online. There's Tree Snap as well, but without the actual leaves where they can look and see if it is an American. We're always interested. Let me just finally say, if the nuts are big, you can't see my fingers.
Yeah, we can.
If the nuts are big, it's probably a Chinese. If the nuts are little, then it might well be an American. So if the nuts look like the ones that you buy in the store at Christmas, which by the way are mostly imported from Italy, then it's probably a Chinese or some sort of hybrid, some sort of, what do they call it? An orchard tree that's been bred for nut production.
Shannon also added, you talked earlier about the little orchard that they have. The chestnuts on the on the Heritage Conservancy Fuller Purcell Nature Preserve were installed as part of the American Chestnut Foundation program to test for blight-resistant trees. There is a local volunteer who monitors them.
Mm-hmm.
And then Ming Wong, who is one of our faculty members here at DelVal. American chestnut tree is monaceous, meaning separate male and female flowers growing on the same individual tree. And now I'll get to some Q&A here too.
Can I just say you still
sure.
need 2 trees? That's my understanding is a tree does not successfully self-pollinate and produce nuts. You need some other tree. You need a female and a male tree.
We had a question early on from Phoebe. I hope this got answered. But what do the cankers on trees look like? I think you showed that in your slides. Phoebe, if you still have questions about that, would you send in a new question related to that? Kelsey has a question. If the tree or if the new bred transgenic chestnuts are put in the forest, will be mixed with the old root system, or will we still see non-resistant sprouts from the old system that continually die off?
Yes, you will still see the root sprouts. And because most of those sprouts don't get to enough size to actually flower or fruit, then the challenge will be to gather the germplasm from that and then somehow introduce the blight-resistant trees back into the forest. I don't know if that answers it, but and there's always chance that some American will find a hole in the canopy and live for 20 years and not have, maybe be blighted, but still flower and fruit, which does happen. And, if there's a transgenic tree near it, then they could, cross-pollinate and squirrels and all take the seeds and bury them. And then the next thing you know, some of those seeds grow and the tree is actually blight resistant. So.
Also from Phoebe, we have, how do I identify a chestnut burr seed from other bird seeds? Look for the leaf and twig bud arrangement, question mark, which I think, again, you dealt with in the presentation. I don't know if you want to add anything.
Now the burrs are just, they're big. They're the size of a softball and they're covered with spikes and they're really prickly. And inside it, there'll be 3 little chestnuts. If the chestnuts haven't been scattered, if the burr opens and once it drops to the ground and opens, then chances are wildlife will have gotten the seeds before you do. So.
And another question from Kelsey. Also, when do you think the transgenic chestnuts will be allowed to be grown in the wild?
Well, that's what we're waiting for, the USDA's approval in the regulatory process. And then it actually has to be submitted to the EPA. And then it has to be, the application then goes to the FDA. Because I believe, because chestnuts are a food source for that humans can eat.
Yeah.
So anyway, I forget why it has so many regulatory hurdles. I do want to point out, there's a lot of genetic modification that goes on in food crops. We all know that. I mean, corn is classic. Tons and tons of it has been genetically modified. Trees are really complicated. They are really hard, much harder to breed, and the genome of the American chestnut has like 40,000 genes on it. Like, it's really big, and it's just not like what plant scientists can do with various food crops and that they deal with, which, by the way, you can grow crop every year, twice a year, whatever it might be, and easily determine what you have. So.
Yeah, if I remember correctly, the Federal Registry had the listing for this for the USDA, right?
They had the open comment period.
That's closed now, hasn't it?
It has closed. It closed, I think it was in October, it closed, and it was 2 months that it was open, and the 63% of the comments were in favor. Now, you could go in and read the comments, and they're tallying the comments. A lot of the comments that were negative were like identical. Like, I think the transgenic tree is a bad idea. Like, and you'd have like, hundreds of people. They got something like 4,600 comments, but there's sometimes repetitive comments. So that might be an organization or whatever says to their members, nothing can be, you can't have genetic modification in anything, and we're against this, and this, and then they would all post the exact same single sentence that says they oppose it. So I just want to also point out that modern medicine relies on a lot of genetically modified substances. People, I think the diabetes drugs they've used some genetic modification. Anyway, it's pretty pervasive in our world. It's very hard now to not accept some of this. Doesn't mean it shouldn't be reviewed and regulated.
Right.
And I'm telling you my perspective on it. But anyway.
Mary, are you aware of any other tree species in which transgenic has been introduced into wild populations anywhere in the world?
Oh, no, this is the first.
Yeah.
This is sort of the pioneering case. Now, I mean, one of the issues is that, like, take hemlock woolly adelgid, that's a bug. That's a different vector for the disease. Emerald ash borer, again, an insect, a different kind of vector. It jumps from tree to tree, kills the trees. So there's other, with a lot of these threatened species, there are different kinds of challenges. But I do believe that there are some opportunities for species restoration, but I'm not versed in that. I can't really speak to it. So, but that's a good question.
I for one would say that, it makes perfect sense to me. I am a conservation biologist. If you would bring up to me transgenic for pretty much anything else in the world for restoration in the wild, I would probably initially have a mini stroke and then would slur a bunch of profanity. But in this particular case with the American chestnut, not, as you said, being a keystone species and having been so very important to a whole host of wildlife species, to the forests all along the Appalachian Mountains, and the impact that it had even on economies for people. This is certainly, I think, a species that is very deserving of all the attention it's getting.
Yeah, there's actually a lot of, philosophical debate about the importance of a wild type American tree genetically in the forest. Think about the back crossbreeding program. Our trees range from like 55 to 90% American, and the rest is Chinese. It's not, that's not a locally adapted, genetic source, and it doesn't, and it looks different. The Chinese tree doesn't look the same as the American, and the way it grows, the way it branches and things like that.
Yeah.
So, you can say to yourself, well, what makes a partially Chinese tree somehow, quote, better than a transgenic tree that has the American genes?
Right.
So
we do have another question from Michael. We were lucky enough to get 1-year-old saplings grown from the seeds of 2 adjacent American chestnut. We planted 25 of these saplings. The parent trees do have the disease, but are still producing. How old do our babies need to be to safely remove leaf sets to test their genetic purity?
Sarah Fitzsimmons at the American Chestnut Organization .org, TACF, she could answer that. I apologize. I don't know that.
So that's Fitzsimmons?
Sarah Fitzsimmons. If you go to the acf.org website, it is you'll find all of the list of staff and everybody there. They'll be able to answer those questions. They've got fact sheets on growing chestnuts and everything. Oh, by the way, I neglected to mention the fact that deer love eating chestnut trees. You saw the picture of the Heritage Conservancy's planting and their tree tubes. You got to have deer fencing. You can plant all the chestnut trees you want, but you better protect them in their young age from deer, otherwise you won't have a tree. So, but anyway, acf.org, and Sarah Fitzsimmons, Director of Restoration, is always happy to answer technical questions. And there are other people as well in the organization who can do that.
Great. And we're getting close to running out of time, but we have one more question from Jack. Can you talk about how many barns and cabins were built with chestnut?
I don't know. I will say that chestnut and oak are often confused, and you really need to talk to people who know wood and building and lumber and all, and how you can identify when you see a timber in a building, is it chestnut or is it oak? I do know, I read somewhere along the line that after the chestnut trees started dying, a lot of the housing that was built after the First World War. Places like Philadelphia, West Philadelphia, all those row houses and all, a lot of them were built in the teens and 20s. And a lot of the trim is, I understand, is chestnut. Typically, that trim was painted. Chestnut, although they use it for furniture, it tends to be more utilitarian day-to-day furniture, not fine furniture. It's not like cherry or mahogany or something like that people used, because it does have a pronounced grain. So anyway, that's all I know, is that there's a lot of chestnut house trim out there, and probably other portions of stick-built houses.
Shannon, are you aware if any of the properties that Heritage Conservancy has, whether there's any uh, chestnut barn boards out there?
I am not sure. I just made a note to ask Jeff Marshall, our barn expert president, because I'd be curious to know if he can tell. I mean, he's got quite the eye for attention to detail and all things barn and historic. So he may very well have some buildings that he can point us to.
Okay. Well, I'm gonna wrap us up here. Mark also had put in the Q&A. He said, "Great presentation. Thank you for your work." And I echo what Mark had to say. And thank you so very much for your work within this volunteer organization and trying to give voice to preserving this tree that we lost and don't want to lose forever. We want it back. So thank you
Yeah.
so much.
Well,
And thank
thank
you,
you.
everybody, for coming tonight. Good night, everybody.
Right. Thanks.